Oh, I guess, there we go. Oh, alrighty, so hello everyone. My name is Dylann Sikstrom, and for the last 4 months, I have had the great pleasure of completing my co-op here at the Wishart Lab Group as part of the engineering group. So today I'm going to be talking about one of the projects that I worked on. It was a very new project as it is more related to, as you can see, like blood collection, which is very out of discipline for what I thought I'd be working on as an engineer, but I've learned a lot and I'm excited to share it with you. So the motivating idea behind the project that I was working on was looking for a way of eliminating inpatient blood collection for clinical diagnostic testing. So blood collection for diagnostic testing is typically done in person by trained professionals, and this makes it very inconvenient and inaccessible for a lot of people, especially for diagnostic testing that requires a small volume of blood. And so if there was a way that we could make this more accessible to people, it could potentially become a more regular thing for people. So the blood collection process, as it's typically done, is blood is drawn from the patient in person at a clinic or hospital. That sample is then sent to the lab. Once it gets to the lab, it is then centrifuged, and from there, the serum can be extracted, and finally, that serum can be used for analysis use. But what if that weren't the case? What if there was a way in which by the time that sample gets to the lab, it is already serum and essentially ready for use? And this is kind of the main idea behind the project. So the project pitch was an at-home blood collection and separation device. So using the help of a device that is already on the market called a Tasso. This company designed this device to help actually with this decentralized clinical trial. So it allows users to collect their own capillary blood, and they have Tasso devices that are capable of extracting around 250 to 500 microliters. And it's a very simple process, does not require any formal training, just follow a couple of simple steps and they're able to do it themselves. So the idea was, what if we could build our own microfluidic device that we could attach onto a device like this? And inside that device, it was capable of, at a highly efficient rate, of filtering the blood from the whole blood, from the red blood cells and the serum. And then what if that serum could be stabilized or absorbed into a material that it could then be shipped to the lab, and once it gets to the lab, it is extracted. So half the work is already done by the time it gets to the lab. So in order to do this, one of the main things was, well, how are we going to actually separate the blood? So there's there's essentially only one way that this is done, which is a mechanical method, and that is the centrifuge. So using centrifugal force, it just forces all the heavier components to settle at the bottom, and the lighter components remain above. And in your tube, you get a breakdown of serum, platelets, and red blood cells. And like I said, at the lab, they can then extract that serum and it's ready for use. So looking into alternative forms was kind of the first step. And alternative forms of centrifuges are kind of these at-home devices that you can make yourself. So you can see that first image there. You basically just pull on those two handles and it creates a rotation. And although they kind of do work, the consensus is that they're very inconsistent. Same with this fidget spinner design. So, and also in the context that we wanted to use it, this wasn't going to be, it couldn't be a small device. So that, of course, that wasn't going to work. And then the other thing is blood kind of naturally does separate on its own. So you can use tubes that have a anticoagulant coating in them. And if you put that in the refrigerator and let it sit for 8 to 10 hours, the heavier components in that blood, so the red blood cells, will naturally settle to the bottom, and then you can extract the serum. But once again, for the concept of this device, that won't really work, because how would you get it out? Would users have to do that themselves? So kind of had to think about how else we could do this. And so that's kind of where the idea of a passive separation method came into light. So what if we had this microfluidic device that could passively filter the blood into its serum component? So the biggest constraint of this project is the fact that at the absolute most, a Tasso device can only extract 1 ml of whole blood, and whole blood is only made out of about 40 to 50% serum, which means out of 1 ml of blood, we'd only get around 500 microliters. And for the diagnostic testing, the goal was to get around 100 to 150 microliters of serum, which means every step in this process had to be really efficient. So this is kind of the project that the steps of the project that I began working on, and I'll go through actually some of my experimental results. So the three key aspects of what my research focused on was filter materials. So that was the first step, is what kinds of materials are actually going to allow this passive separation to happen? What kind of material could we put underneath that filter that will either help with that separation by potentially actually helping and pulling the serum through the filter? How will these materials actually store the serum? Will it be stabilized in a way that it can be shipped? And finally, once we get the serum in there, how can we get it out? So these are the three sets of experiments that I'm going to show next. So the first thing that I worked on was the filter membrane. So this was actually testing if there was a filter out there that had the capacity to filter the red blood cells from the serum. So we tried filter paper, which is just kind of the standard lab filter paper that's used. The problem with that one, it was very absorbent, and that you'll kind of see going through. I use it as our control, because if it can filter it but is very absorbent, can we find another material that does the same but absorbs less? Tried polyester membranes of various sizes. Also tried a cell strainer at a much larger pore size, and then also tried this membrane here called a Pol Vivid plasma separation membrane. So it's actually a product on the market that is directed towards this type of use. And also tried dialysis tubing, because it's naturally a semi-permeable membrane that blood is meant to flow through, so it's got to work, right? So this is some of my initial testing where I used a very small volume of blood. The idea here was just to get a general feel for if any of these filters had potential. So I just had an O-ring set up that had the filter placed inside of it, and then one of the absorbent materials that I'll get into more later underneath it, so that we could see if there was any evidence of a liquid coming through. So you can see right off the bat that filter C, the pore size was much bigger than it should have been, and it did leak right away. So did not continue testing with the cell strainer. But you can really see the results in these pictures featuring the PEG. You can actually see you can actually see where the crystals there have become wet and they've become this more clear opposed to the white, and that was actually the best evidence of serum coming through the filters. And you can even see when it got really wet that it kind of like clumped together in this picture B here, showing evidence that serum was in fact coming through these filters. So after lots of testing with larger volumes of blood, it was determined that the polyester membranes and the plasma membranes were working the best. Dialysis tubing was one that took a little bit longer to rule out. It is naturally a wet membrane, which made this process a little bit trickier, because no matter what you coated around it to absorb the serum that potentially came out, it was always wet, because it was just absorbing like the natural moisture from the filter itself. But after quite a bit of testing, it was determined that no serum was actually coming out. So I tried it as a dry filter membrane, so just cutting like a little circle out of it, using it like the other ones. I tried that same method, but treating it as the wet membrane that it should be, and then also treating it as a tube filter, as you can see in those pictures there. And unfortunately, none of those showed conclusive results of it actually filtering out any serum. So also moved on from the testing of that one. But in the end, this was very successful, because found about 3 filters that were capable of filtering serum out of whole blood. So the next step was then looking at absorbent materials that could be placed underneath these filters to, like I said, absorb the serum that comes out. So numerous things were tried here. Once again, tried that lab filter paper, because it is naturally very absorbent. tried a specialized polymer, tried PEG, and also tried microcrystalline cellulose. And I will show you the results on the next slide. So one of the testing that immediately got ruled out was the microcrystalline cellulose. As you can see, I don't have any pictures of that experiment because it was just very clear right off the bat that it did not work, as it was very similar to the experiments as before, and there was no evidence of the microcrystalline cellulose underneath the filter being wet. So continued with the other stuff. So these are the results of the filter paper. So I just made a little device basically to hold my filter in place with the filter material, because one thing from my testing that I learned was the contact between the absorbent material and the filter membrane was really important for higher volume yield, because if there wasn't contact between those two, essentially nothing was coming out. So that was the purpose of this device. And out of 125, so I added 500 microliters of whole blood to each of these tests you see here, which means, or sorry, my apologies, I added 250 microliters of whole blood for each of these, which means that there would have been 125 microliters of available serum. And from just doing a weight test of before and after, it was determined that the most amount of serum that came out of any of these filters with the filter paper was only 28 microliters, which would not have been enough. And another interesting thing that we learned about the filter paper is if you take a look at the color of the filter paper, if this was just serum that was coming out, it would be like a yellow color at most, but you can see it's very pink, almost red in this case. And so something interesting was that the filter paper from underneath the filter material was somehow causing hemolysis of the red blood cells, and we were actually seeing this color, which indicates that those red blood cells were breaking. So that was very interesting. And then the materials that worked the best by far were the polymers. So you can see here, I have both shown the specialized polymer and the PEG, and they both had the best ability to pull the serum through the filter and actually absorb it. So with the specialized polymer, you can see on that bottom picture, you can actually see the serum just sitting on the bottom side of that filter as it gets absorbed by that polymer. And same with the PEG, you'll see the redness, and that's just the blood on the other side of the filter, but none of that actually came through the filter. So you can actually just see how I was just able to wipe it off. And that was kind of the deciding factor between the two, is that the PEG was just easily wiped off, whereas the specialized polymer had this tendency of embedding itself into the filters, which would make it less efficient overall. But we still tested extraction methods for all of these materials. So the only one that I did not, the only material here that I will not be focusing on is the filter paper, because the lab already has methods for those. But the other three absorbent materials, we did run tests on to see if there was a way of extracting. And so this has kind of been what I've been working on most recently. So this is still in progress. This was completed by Abby, so thank you for that. But attempted to actually dissolve the specialized polymer using a variety of salts. Only one of them ended up working, so you can see as the concentration of that salt increases, you can see the polymer begin to clump together, and the idea was that it could then be removed and the serum would be usable. We are still working on testing these and determining if that is the case. But we also tried this salt test on PEG, and unfortunately that was unsuccessful in dissolving anything. The next material that we attempted to extract from was microcrystalline cellulose. So it's just a powder, and the idea was as you add more serum to that, or sorry, as you have more of that microcrystalline cellulose and you add more serum to it, it becomes like a thicker paste, which is really good for shipping and all of that. But the problem that started happening was the thicker pastes were harder to extract from, because obviously that concentration is higher. So I wanted to try about 3 different types of concentrations and see if we could extract anything from it. So the way that I tried to do this was centrifugation, because the idea was that those microcrystalline molecules would sink to the bottom and your serum would go to the top, similar to how it does in whole blood. And you can see here that is more or less what happened. There was 250 microliters of serum added to each of these tests, and you can see that the most amount of serum that was extracted was only 150 microliters. So these results were okay, but they weren't the best that we'd had so far, and you can actually see the interesting thing was, as you can see, that the cellulose around the tube after centrifugation did in fact return to its powder form. So it was working, but it still didn't work quite well enough. So continued to focus on, can we extract from PEG? So did a very similar thing as the microcrystalline cellulose. You can see I tried 4 different types of PEG. So these are all just different molecular weights. The idea here was that the higher the molecular weight, the better the centrifugation would work, because, like I said earlier, those heavier molecules sink to the bottom and your serum would stay sitting on top. So similar to the last test, tried the same 3 concentrations, and these are the results. So this slide is a little bit overwhelming to look at first, but we'll just first look at the 8,000 PEG. So you can see that at first it's very milky in color, but you can see after only 5 minutes of centrifugation, you can see that that clump of PEG begins to form at the bottom, which is exactly what we were looking for. And as the time goes on, that clarity is brought back to the serum. You can see a similar thing happen with the 25,000 PEG. Once again, you see that clump form at the bottom, and you can see the clarity increase, which were all really good signs. But unfortunately, you can see for the higher molecular weight PEGs that there's a clump forming, but they're still very milky. And these are the ones that we thought would work the best. But one thing that we didn't anticipate was the higher molecular weight PEGs actually made much thicker mixtures. Despite them being the same concentration, they were almost like they almost cemented, that's how thick they were, which would just ended up being too thick to centrifuge. So despite the fact that those bottom 2 do look like the PEG was coming out, if you actually flipped them upside down, they were still so thick that the liquid inside wouldn't move. So from all of that testing, I was able to extract serum from only 5 of those. And like I said, this is still testing that I've been working on, so we've yet to actually determine if the serum is left fully intact after that, but that's just another step that will happen in the future. So kind of like to wrap everything up, the concluding results are, yes, there are filter materials that can successfully passively separate blood. There are absorbent materials that can absorb that serum. And like I said, the extraction methods are still a work in progress. But with this project, I was able to learn so much and just try my hand at a lot of laboratory work that I never thought I'd get the chance to use. And now they're building blocks for, I think, a really great project. So that is the end of my presentation. Thank you for listening. And like I said, thank you to everyone who has helped with this project and who I've had a chance to work with. I really enjoyed my time here. So thank you.